This presentation includes our research project on hypoxia and endothelin-1 in microglial cell activation. Studies have proven that endothelin-1 production is stimulated in the onset of hypoxia. However, the role of endothelin-1 in microglia under hypoxia is not clear. Therefore, we hypothesize that if endothelin-1 in the onset of hypoxia promotes pro-inflammatory properties of human microglial cells, then a blockade of the endothelin-1 receptor will reduce its pro-inflammatory response. Our aim is to assess and clarify the effect of hypoxia and endothelin-1 in human microglial cell line HMC3. The clinical relevance of this project is tied to ischemic stroke. In the United States, the incidence of stroke is close to 800,000 cases per year, of which more than 85% are of ischemic nature. Literature indicates that hypoxic states trigger microglial cell activation and proliferation. Now, in the onset of ischemic stroke, dual microglial cell activation is required in order to properly address debris removal and wound healing processes. Also, a rise in endothelin-1 has been identified in the onset of cerebrovascular disease. This project was organized in two parts. In the first part, we compared differences with regards to the pro-inflammatory response of HMC3 grown in normoxic versus hypoxic conditions. To induce hypoxia, we placed cultures in a gas chamber of 1% oxygen, 5% carbon dioxide, and 92% nitrogen for 4 hours. Immunofluorescence staining was performed after hypoxic treatment, and measurements of qPCR, ELISA, and Muse oxidative stress assay were performed 0 hours, 24 hours, and 48 hours after treatment. In the second part, we compared differences in pro-inflammatory state induction of HMC3 cells following the addition of exogenous endothelin-1 and endothelin-1 receptor antagonist BQ788. Here, HMC3 cells were grown in normoxic conditions, and experimental groups were treated with ET1, BQ788, and ET1 with BQ788 combined. Measurements following treatment included flow cytometry, ELISA, and Muse oxidative stress assay. As it can be seen in the image, immunofluorescence staining clearly indicates an increase in ET1 gene expression within HMC3 grown in hypoxic states when compared to their normoxic counterparts. Gene expression of ET1 was further quantified by qPCR analysis, where ET1 expression was markedly increased in cells that underwent hypoxia, independently from the time of measurement. This includes an initial 5-fold increase immediately after hypoxic treatment. In addition to ET1 gene expression, ELISA measured an initial 1.3-fold increase of ET1 protein products in hypoxic HMC3 cells when compared to their normoxic counterparts. Also, the levels of pro-inflammatory cytokines, TNF-alpha and IL-6, increased by factors of 3.8 and 1.6 respectively. These values remained increased after 24 and 48 hours of treatment. Moreover, reactive oxygen species production increased in hypoxic HMC3 cells by a factor of 2.5 and remained increased by measurements following 24 hours of treatment. Now, these images correspond to changes in normoxic HMC3 cells treated with exogenous ET1 and ET1 receptor antagonist. In the flow cytometry to the left, we can see that HMC3 cells of anti-inflammatory phenotype, which are represented by M1, predominate in the onset of controlled normoxic conditions and remain unaffected by the addition of the ET1 antagonist. However, administration of ET1 induces the activation of the pro-inflammatory phenotype, leading to an increased M2 count, and addition of the ET1 antagonist drive those levels back to baseline. Furthermore, exogenous ET1 administration increases reactive oxygen species production by a factor of 1.4 in normoxic HMC3 cells, and this increase is blunted following administration of ET1 antagonist. Finally, exogenous administration of ET1 to normoxic HMC3 cells increased the levels of TNF-alpha and IL-6 by factors of 1.6 and 1.9 respectively. These increases were inhibited following administration of ET1 antagonist. In summary, hypoxia leads to an increase in endothelin-1, pro-inflammatory cytokine, and reactive oxygen species production in HMC3 cells. Exogenous endothelin-1 administration induces the HMC3 pro-inflammatory phenotype and response, and endothelin receptor antagonist inhibits HMC3 pro-inflammatory state progression. In conclusion, this project confirms that hypoxic states induce microglial cell activation, upregulating the production of reactive oxygen species and endothelin-1. Also, it was proven that endothelin-1 further stimulates microglial cell activation, creating a cycle of microglial pro-inflammatory state progression. And finally, endothelin-1 receptor blockade could present promising therapeutic effects in regulating microglial response in hypoxic ischemic brain injury.